Assalamu alaikum. Good afternoon, good evening, or good morning to all of you who are joining us from different parts of the world. I'm Dr. Sohaila Chima. I'm the Assistant Dean for the Institute for Population Health here at Weill Cornell Medicine, and also an Associate Professor of Clinical Population Health Sciences. So a very, very warm welcome to each of you joining us today for the Population Health and Wellbeing series. As many of you know, the objectives of this series are to discuss contemporary and critical topics which are relevant to healthcare, medicine, and population health. We also examine evidence-based practices which are germane to public health and patient care, and also then talk about opportunities and challenges in the evolving face of healthcare and population health. So just some housekeeping. Your cameras and microphones have been turned off. You can use the question and answer feature, the tab, to type questions, comments. You can actually also use the chat feature. Just put in your comments there, anything that you want to, we will be able to view them. And again, I will be moderating the questions at the end of the session. Again, as many of you know, this is an accredited activity, and it is accredited both by ACCME and also by DHP, which is our local accreditation body here at the Ministry of Public Health. Evaluation and certificates. The post-activity evaluation, it will be available in your Cloud CME account by October the 23rd. You will receive an email notification once that evaluation is available in your Cloud CME account. And certificates will be available for download once you complete the post-activity evaluation on the Cloud CME portal. Now, coming on to the speaker today, I'm very, very excited about this topic, and it gives me great honor to introduce Dr. Liz Arbittier. So she's going to be talking to us about One Health, the contribution of veterinary medicine in transforming concept into practice. So Dr. Arbittier, she's an Associate Director of Academics and Community Affairs for the Department of Clinical Studies, New Bolton Center at the University of Pennsylvania School of Veterinary Medicine. She received her Bachelor's of Science degree from Drew University in New Jersey, and then received her veterinary degree from Penn Vet in 2001. Thereafter, she entered private equine ambulatory practice with a focus on equine sports medicine. Dr. Arbittier returned to New Bolton Center, a large animal hospital at Penn Vet, as a member of the Equine Field Service team in 2013 and moved into her current role in 2022. Through her service in national committees, her long-standing role on the Penn Vet Admissions Committee, and her task force on recruitment and retention of equine veterinary students at Penn Vet, Dr. Arbittier is currently focused on helping to improve the critically low numbers of equine veterinarians being produced in the United States. Dr. Arbittier, very, very warm welcome to you. We're really excited that you could join us today. Over to you.
Thank you so much, Dr. Chima. I'm thrilled to be here. Thank you, everyone, for joining us. This is an interesting topic. I hope you find it interesting as well. So, as stated, I'm Liz Arbittier, and I'm thrilled to be here today to discuss the contribution of veterinary medicine to One Health. This is material that could very easily fill a full day of talks, so I'm going to give you a bird's eye view of some different areas of One Health and how veterinary medicine is contributing generally and then specifically at the University of Pennsylvania. One Health is a very hot topic at most veterinary schools, and I can't stress enough how deeply it's rooted at the core of the mission at Penn Vet. I have no disclosures, and you should have all received the learning objectives. Okay. On May 29, 2007, a family physician in a rural village in the south of the Netherlands in the Noord-Brabant province alerted the regional public health service because of an unusually high number of cases of atypical pneumonia in his adult patients. Soon after, another family physician in the same area made a similar alert. At the time, they thought maybe they were dealing with bacteria pneumonia caused by Mycoplasma pneumoniae, which was a common cause. But additional serological tests rapidly led to an identification of an outbreak of acute Q fever, the causative agent of which is Coxiella burnettii. Q fever is an infectious disease that affects both humans and animals. The Q comes from the word query. It was called query fever until its true cause was discovered in the 1930s. C. burnettii is a pleomorphic coccobacillus, referred to as being gram-negative, although it stains relatively poorly with gram stain. It is an obligate intracellular pathogen in nature, and within the cells, the bacteria replicate inside vacuoles, which is what you can see here. And it has some properties that become important to what make it a dangerous threat. During the course of its development, there are two morphologically distinct forms of C. burnettii. The large cell variant, which is a vegetative form found in infected cells, and the small cell variant, which is the extracellular infectious form shed in milk, urine, vaginal secretions, semen, and feces, and found in high concentration in placental tissue and amniotic fluid. The small cell variant is resistant to heat, desiccation, and many common disinfections, and it remains viable for weeks to years in the environment. After infecting a domestic ruminant, C. burnettii can localize in mammary glands, supramammary lymph nodes, placenta, and uterus, from which it can be shed in subsequent parturitions and lactations. Transmission can occur via inhalation of dust that has been contaminated by infected animal feces, urine, milk, and birth products. Direct contact, like touching and being licked with an animal, is not required to become sick with Q fever. People also can get Q fever by eating contaminated, unpasteurized dairy products. Seroconversion is asymptomatic in 60% of cases. C. burnettii actually fulfills all of the requirements to be considered a biological weapon. It consistently causes disability. It can be manufactured on a large scale. It remains stable under production, storage, and transportation conditions. It can be effectively disseminated via aerosol, and it remains viable for years after dissemination. C. burnettii is incredibly highly infectious, in some cases requiring less than 10 bacteria to create clinical signs. And as I said, it's very resistant to heat, drying, and common disinfectants. It's deemed a Category B biological terrorist agent since, although it has potential for large-scale dissemination easily, it lacks capacity for the massive fatalities potentially caused by Category A agents like smallpox, anthrax, plague, botulism, tularemia, and viral hemorrhagic fevers. If used against troops, it's thought that C. burnettii can cause a loss of manpower that can range from 23 to 77%. And if used as a biological weapon in a civilian population, the degree of infectivity can actually rival that of anthrax. And although associated mortality is low, it will cause extensive acute and chronic morbidity. The acute symptoms of Q fever are headache, fever, malaise, muscle aches, abdominal pain, nausea, vomiting, fever, chills and sweating, loss of appetite, and diarrhea. In this outbreak, fever was the most frequently reported symptom at 90%. Malaise and headaches ran from 63 to 92%, coughing in 66%, night sweats in 80%, and 66% of them had pneumonia. Hepatic insult, which is also seen, was less common than in previous reported outbreaks at 15%. In this particular outbreak, the hospitalization rate was significantly higher than previously reported. In 2007, 50% were hospitalized, and in subsequent years, 20% were hospitalized, compared to just 2 to 5% in the literature. As predicted, the death rate was low. Out of 749 hospitalized patients, only 9 died, and all those who died had pre-existing comorbidities. That's a death rate of about 1%, and you see generally 0.5 to 2.4% death rate reported in the literature. Chronic Q fever looks a bit different. It can create valve lesions or endocarditis, chronic fatigue syndrome, osteomyelitis, chronic hepatitis, interstitial lung disease, and repeated abortions. Despite the implementation of measures aimed at identifying and controlling the source of infection, the number of acute Q fever cases notified to Dutch healthcare authorities did not stop increasing. And as you can see from this slide, from 2000 to 2006, only 5 to 20 cases per year were reported. In just July of 2007, there were 63 cases reported, 59 in that particular province. In all of 2007, there were 168 cases. In 2008, 1,000 cases. 2009, 2,054 cases. Still elevated in 2010, and then in 2011 and 12, it started to come back down. The Noord-Brabant province was the most affected region, and it has about 2.4 million people and 6.4 million farmed animals, mostly pigs. Goat farming is a marginal agricultural activity in the Netherlands from an economic point of view compared to pig, cattle, and poultry farming, but it is concentrated in the south of the Netherlands where this province is, with some of the world's largest goat farms existing. So you can see they had over 4,000 cases reported in 3 years. This started a massive public health investigation. I don't actually believe any guns were involved, but this was just a cute graphic. You can ignore that guy. One of the first tasks of the Dutch public health authorities was to try to identify the source of infection in order to control it. The source of infection was clearly identified for some of the clustered cases, like, for example, an orphan lamb was adopted by patients in a psychiatric institution, and it was the cause of 28 cases of acute fever in that institution. Public visits of a farm on lamb viewing days was probably the cause of infection in about 146 visitors to that farm. And there was an urban cluster of 96 patients that allowed identification of a single dairy goat farm as a source. That said, there was no single source identified for this huge outbreak. And so the hypothesis of multiple sources was raised during the first epidemiological investigations. Farms raising small ruminants, and especially dairy goats, were rapidly identified as the potential sources of the human outbreak. Collaboration between veterinarian and human healthcare authorities was required to implement control measures for the outbreak. The regulations concerning the management was greatly modified in the spring of 2008. So you can see in June of 2008, mandatory reporting of abortions was put into place. In October of the same year, animal vaccinations were available on a voluntary basis. In February of 2009, stricter hygiene measures were put into place. And in April of 2009, animal vaccines were mandatory if there were greater than 50 goats or sheep in the affected area. Unfortunately, these measures were insufficient at controlling the human outbreak, and some drastic measures were implemented to eradicate the bacterium. Screening for C. burnettii in farms became systemic with the implementation of continuously repeating DNA testing in their bulk milk tanks. Then it was decided to systematically cull gestating ewes and goats on infected farms in December of 2009. This led to the culling of more than 50,000 animals on 88 farms. We're going to come back to that number. The decrease that followed in human cases was primarily believed to be due to the effectiveness of the veterinarian and physician measures, but also thanks to the increased immunity of the exposed population. Interestingly, they monitored the workers who were culling the animals, and out of the 517 workers, despite the use of protective personal equipment, 17.5 of them still seroconverted for antibodies to C. burnettii. They were tested before and after the culling, and 31% of the seroconverters were symptomatic, and this was probably underreported. So this is just a great case of One Health in action. Physicians and nurses working with lab doctors and staff. Many governmental agencies were involved. Small ruminant veterinarians were critical. Wildlife veterinarians were involved. Field environmentalists, epidemiologists, and the farmers and the farm workers. The wildlife veterinarians got involved because they were surveying animals who lived locally. And in one research study, they looked at roe deer who were found dead or hit by car, and they were surveyed for C. burnettii, and C. burnettii was found in 23%, nearly a quarter. But interestingly, the two strains identified in the deer were not the same as the predominant strain involved in the Q fever epidemic. So it may just exist in the wildlife and not be spilling over into humans. The field environmentalists and ecologists noted that Q fever notification was correlated with environmental conditions, particularly wind current and humidity being the two most significant factors. And 10 years later, the government launched a screening program. They targeted people with risk factors in previous outbreak areas. They found 9 people testing seropositive who were thought to have chronic Q fever, and it was ruled positive as chronic Q fever in 4 cases. All right, so that was just a case to introduce to you the idea of One Health in action. But what is One Health? And you guys had some great ideas in the chat earlier. One Health is the collaborative effort of multiple disciplines working locally, nationally, and globally to attain optimal health for people, animals, and the environment. The mission of One Health is the establishment of closer professional interactions, collaborations, and educational opportunities across the veterinary and medical professions, together with their allied sciences, in order to improve public health and animal health. And today I'm really here to talk about the veterinarian's role in One Health. So I'm going to give a very brief history of One Health. In the 19th century, a human physician, Rudolf Virchow, coined the word zoonosis, and he wrote, "Between animal and human medicine, there are no dividing lines, nor should there be. The object is different, but the experience obtained constitutes the basis of all medicine." This was followed in 1947. Dr. James Steele, who's a veterinarian and also a master in public health, he founded the Public Health Division at the CDC. And in 1966, Dr. Calvin Schwab, also a veterinarian, master in public health, coined the term One Medicine. He worked at the University of California, Davis Veterinary School in the United States. In September of 2004, a group met in New York City and formulated 12 Manhattan Principles that called for the international community to adopt a holistic approach to combat threats to the health of life on Earth under the banner One World, One Health. It was organized by the Wildlife Conservation Society and hosted by the university. Representatives from all of these groups were among the many participants. And we don't have time for me to go through the Manhattan Principles, but 7 out of the 12 directly mention animals, and the animals are indirectly involved in all of these principles. This is just a very simple Venn diagram showing One Health as the intersection between animals, humans, and the environment, with all of these areas being involved. Truly, the key word is multidisciplinary. So in our case, we discussed a One Health response to a zoonotic disease that also impacted food security and public health policy. These are all challenges that benefit from a One Health perspective, and we're going to touch briefly on most of them. I'm going to spend a little more time just talking about zoonoses, as it's such a huge part of One Health. All right, we will see if I can get this video going. Let me know. Oops. Hang on. Let me get back to that. This worked so well in practice. right. That is a short video on the Penn Vet Institute for Infectious and Zoonotic Disease. Because like I said, at Penn Vet, we are very, very invested in the One Health movement. So much of the recent emphasis on One Health has been on pandemic infectious disease, acknowledging that environmental changes like deforestation, climate change, incursion of people into wilderness areas precipitate the spread of diseases originating with wildlife to domestic animals and then to human populations. Outbreaks of Ebola, MERS, SARS, H5N1, and H1N1 infections have focused attention on the interaction between wildlife, production animals, and humans, and really highlights the need for this combined medical and veterinary approach to address these challenges. And in this day and age, we can't discuss zoonotic disease without touching upon COVID, because we have now lived through a historically catastrophic pandemic, and we need to think about how we got there. If you query COVID and One Health on PubMed, you get nearly 37,000 results. The goal of the One Health strategy is to prevent and control the emergence, the reemergence, or the dissemination of zoonotic pathogens. We need to be surveying animal reservoirs, looking at factors facilitating emergence, and taking action for disease control. In an optimum One Health approach, surveillance in the Southeast Asian bat species harboring coronaviruses similar to SARS-CoV-2 would have included regular biobanking from reservoirs and analysis of the environmental factors favoring transmission. Action then could have been taken to control the risk of spillover or transmission to humans. And that, had that happened, we would have prevented millions and millions of deaths, potentially, right? This is interesting. I first obtained this information on October 4th, and I revisited it on October 12th, and you can see the death toll from COVID continues to increase. 6,961,014 deaths attributed, and that doesn't count all the ones that are not reported. This graphic shows the stages involved in a pandemic crisis from the initial circulation of pathogens within wild fauna and the natural environment to the global dissemination after large-scale human-to-human transmission. Anthropological factors lead to changes in pathogen circulation and to increased contacts with new animal species, including domestic ones, which can induce spillover with possible transmission to humans. The COVID-19 pandemic highlights the risk of reverse zoonoses due to intensive circulation of SARS-CoV-2 in humans, which then transmitted it to new animal species, and it circulated within some that were proven to be incredibly susceptible, like hamsters, minks, and white-tailed deer. Cats, domestic cats, and big cats also proved to be susceptible. And at that point, you can end up with a mutated virus being passed back to humans. All right, we're going to move on now to antimicrobial resistance. Antimicrobial resistance, probably you're all familiar with it. It is the resistance of a microorganism, including a parasite, a virus, a fungus, or a bacteria, to an antimicrobial drug that was initially effective for treatment of infections caused by that microorganism. Resistant microbes are found in people, animals, food, and the environment. They can spread between people and animals and from person to person. Antimicrobial resistant genes can spread between microbes, and resistance can occur in the absence of antimicrobials. Why are veterinarians critical at looking at antimicrobial resistance? As human practitioners, you're all familiar with all of these causes of antimicrobial resistance, but maybe you're not familiar with the concept of the current overuse of antibiotics in livestock and fish farming. The development of resistance is often complicated and multifactorial, but farmers are using antimicrobials indiscriminately with their production animals. And they do this to prevent disease in healthy animals. And they also believe that it improves growth and their production numbers, which is not substantiated by facts. The World Health Organization convened a guideline development group, or GDG, of external experts. The GDG assessed the evidence and formulated recommendations that were peer-reviewed by an independent group and approved by the World Health Organization Guidelines Review Committee. In late 2017, to preserve the effectiveness of medically important antimicrobials for humans, the World Health Organization released guidelines on the use of antimicrobials in food-producing animals. These guidelines recommended reductions in the overall use of medically important antimicrobials in food-producing animals, including complete restriction in the use of antimicrobials for growth promotion and for disease prevention. So in other words, in healthy animals considered at risk of infection. These guidelines also recommend that antimicrobials identified as critically important for humans not be used in food-producing animals for treatment or disease control unless susceptibility testing demonstrated the drug to be the only treatment option. Examples of those antimicrobials are quinolones, third and higher generation cephalosporins, macrolides and ketolides, glycopeptides, and polymyxins. Here's a scary fact. In 2019, the World Health Organization identified 32 antimicrobials in hospital development, and only 6 of these were classified as innovative. We are running out of options. This was a letter from the editors in the Scientific American publication just published on March 1st, 2023. This is still a very, very real threat. Factory farms are the ideal breeding grounds for antibiotic-resistant infections because you have a lot of animals in crowded and unsanitary conditions. You add to that the fact that antimicrobials are given at low doses, which kill off the weak pathogens while inadvertently selecting for the strongest pathogens. And that's the perfect storm to create bacterial resistance. In the United States, the chicken industry has actually made some progress, mainly thanks to consumers in the past 10 to 15 years. Farmers used to be injecting chicken eggs with antimicrobials in order to prevent new chicks from catching an infection from microbes on the egg. Now they wash the eggs, which creates the same effect. In the United States, drug-resistant infections killed nearly 5 million people in 2019. 5 million people in 1 year. Many fast food chains have pledged to set reduction targets for the amount of medically important antibiotics in its beef, and almost none of them are hitting the targets that they set for themselves. And recently, an investigation by the Bureau of Investigative Journalism and The Guardian found that companies supplying beef to major distributors are still getting meat from farms using antibiotics classified by the World Health Organization as of the highest priority, critically important to human health. Focusing on antimicrobial misuse from a veterinary perspective is critical. And in the United States, farmers can buy whatever antibiotics they want without a veterinarian's input. They can go to a catalog or go to a store. Not every antimicrobial, but most. This article discusses that in the United States, there's no federal agency that has the power to mandate a decrease in the use of antimicrobials, and efforts to go through our government are likely to be fraught with difficulty. But if we can just regulate the sale of antimicrobials and have it go through veterinarians and have some veterinary input into these farmers using antimicrobials, we could make a huge dent in this problem. And this is a major cause of antimicrobial resistance. All right, switching topics to another challenge that completely benefits from a One Health perspective. I'm sure most people here are familiar with the term translational medicine, but just in case, translational research is where laboratory science and clinical medicine meet to develop novel therapeutics to prevent, diagnose, and treat disease. It applies findings from the study of animal models to improve human health. Veterinarians are uniquely qualified to contribute to this field because of their knowledge of animal systems and their clinical training. Yet veterinarians are significantly underrepresented in biomedical research. In 2014, the NIH actually set off to identify the barriers preventing veterinarians from pursuing research careers. And some of these barriers are that many veterinary school curricula don't encourage veterinary researchers. The heavy load of student debt carried by many graduates of veterinary school, research pays lower than clinical veterinary practice. And in the United States, our veterinarians are graduating veterinary school with easily $300,000 in debt. And then veterinarians are often overlooked when biomedical research teams are being formed. And the report estimated that veterinarians comprise just 3% of the physician-scientist workforce funded by the NIH. At Penn Vet, research at the University of Pennsylvania, actually, in general, research is a huge part of our curriculum. And it is a reason that some people choose Penn Vet and a reason that some people avoid Penn Vet. But research is very deeply rooted in the inception of our veterinary school. Translational medicine in and of itself is a very significant arm of the One Health movement at Penn Vet. These are just a few of the labs working in translational medicine. I'd like to highlight two. In 1974, the Referral Center for Animal Models of Human Genetic Disease, or RCAM, was established at Penn Vet with the overall objective to serve as a national referral and resource center to discover, characterize, maintain breeding colonies, and make available dog and cat models with hereditary diseases homologous to those found in human patients that can then be used in preclinical trials. We also can create large animal models of human disease in collaboration with the Penn Vet Center for Animal Transgenesis and Germ Cell Research core facility, which was established in 1998. Interestingly, scientists at Penn Vet, under the leadership of Dr. Ralph Brinster, performed pioneering studies in the development of transgenic techniques and were responsible for the production of the first transgenic mouse. American President Barack Obama bestowed the 2010 National Medal of Science upon Dr. Brinster for fundamental contributions to the development and use of transgenic mice. This is the highest accolade bestowed by the United States government on scientists and engineers, and Dr. Brinster was the first veterinarian to win the award. Also just a very nice man. The other lab I'd like to focus on is the Animal Model Core and Comparative Orthopedic Research Lab. Under the direction of Dr. Tom Scher, this lab is part of the Institute for Medical Translation at Penn Vet's New Bolton Center. So this exists at the large animal hospital. This group leads, supports, and collaborates on many federal and industry-funded projects intersecting science and the rapid advancements in healthcare technology. The key here is multidisciplinary. People from the medical school, the engineering school are all involved. There's two cores to this lab. There's a service core, which designs and executes studies from proof of concept to preparation for clinical studies, and the research core, which initiates and collaborates with academia on hypothesis-driven research using animal models in support of investigations in the areas that are listed here. Smaller scale studies can take advantage of their large biobank of specimens before moving into in vivo studies. This lab provides opportunities across most common model species like rodents, rabbits, miniature pigs, Yorkshire pigs, sheep, goats, llama, alpaca, calves and cows, and horses. This is just one example of a study. Let me get rid of this here. And that box will disappear. A study that was performed out of this lab, and it was looked at treating large bone defects caused usually by high energy trauma and tumor resection in humans. They used their ovine tibial critical defect model to perform a direct comparison between two implants, a polymeric mesh and a graft cage. Here you can see the timeline for the study. The green boxes indicate radiographic time points, and the orange boxes indicate CT scan time points. At the end of the study, histomorphologic evaluation was performed, evaluating fracture healing and the host response. Torsional testing was performed, in which the tibia was secured at both ends and rotated in a manner that simulated external rotation. They compared results to the torsional strength of the contralateral limb, and finally they compared the two devices. Currently, this lab has 30 funded active projects with an additional 9 currently under review for funding. Taking a different look at translational medicine, if you have an interest and you'd like to get your feet wet, I highly recommend this book, Ubiquity, which won all kinds of awards. One of the authors, Dr. Barbara Nadesan Horowitz, is a cardiologist whose interest in bridging human and animal medicine began after the Los Angeles Zoo called her to consult on an emperor tamarin suffering from heart failure. While examining the tamarin, a veterinarian warned her against inducing capture myopathy, a term with which the cardiologist was unfamiliar, but veterinarians are very familiar with. Capture myopathy is an often fatal exertion or stress-induced muscle degenerative condition affecting captured wild animals. The book is divided into 12 chapters, with each focusing on a human condition alongside its animal parallel. The topics cover a broad range of disease, physical, and behavioral. It's a very easy read, and it is really interesting. Some barriers still exist to One Health And Dr. Mary Christopher, who is a veterinarian PhD professor emeritus at the UC Davis School of Veterinary Medicine in California, United States, writes that translational research has blurred interdisciplinary boundaries, but there are still some boundaries that exist. And one of them is the separation of veterinary and medical research. She discusses the concept of one literature, which extends the One Health approach to how we retrieve, cite, and publish biomedical research, removing contextual boundaries between veterinary and medical literature and facilitating the knowledge exchange and collaborative approaches that benefit translational research. And she really gives 3 distinct ways that we can fix this problem. Indexers need to cross-list veterinary specialty journals under relevant medical specialties. Editors need to draw on both veterinary and medical experts in their reviewer pools to bring appropriate scientific expertise to the critical evaluation of these manuscripts. And publishers need to develop collaborative veterinary medicine ventures, such as joint publications that facilitate the connections between the professions. Just an interesting graph. She pulled research articles involving spontaneous canine lymphoma as a translational model for human lymphoma. And you can see that the translational model papers have tripled since the year 2000. So the work is being done. We're now going to touch briefly on the human-animal bond. And when you're discussing the human-animal bond, there are multiple facets that need to be looked at. Animals serve a vital role as companions and truly as members of the family. And in our case, we also discussed different type of animal bond, human-animal bond, when more than 50,000 animals were culled during the Q fever outbreak in the Netherlands. These mass extermination of animals are extremely difficult for those who have to carry it out, and the impact on the people who have to do this has been widely studied. The American Veterinary Medical Association defines the human-animal bond as a mutually beneficial and dynamic relationship between people and animals that is influenced by behaviors essential to the health and well-being of both. There are health benefits that have been proven to be related to the human-animal bond: reduced anxiety and stress, decreased blood pressure, decreased cholesterol and triglyceride levels, leading to decreased risk of heart disease, asthma prevention for children, support for kids with autism, assistance for trauma survivors with PTSD, increased social interaction, and increased activity to control obesity, to name a few. When humans are separated from their pets in times of crises, their health plummets. And we saw this initially during the huge AIDS crisis in the 1980s in the United States, and also recently for COVID-19. This is just pulled from an editorial by Dr. Michael Day. Pets, we know, bring enormous well-being to their owners, but they also can bring risk. And this is just a nice table of common zoonotic infectious diseases passed on from dog and cats to their families. And so as a human physician, when you're looking at a case, you need to be aware of these zoonotic diseases and what animals that people have in their household. Other ways animals can affect humans negatively are through allergies, animal bites and scratches, and human falls. And then there's also the grief and negative emotions when a companion animal is injured or dies, which is also significant. But animals can help in unexpected ways. The NIH reported on a recent study that showed that caring for fish helped teens with diabetes better manage their own disease. Researchers had a group of teens with type 1 diabetes care for a pet fish twice a day by feeding, checking the water levels, and changing the tank water each week. This was paired with the children reviewing their own blood glucose logs with their parents. Researchers tracked how consistently these teens checked their blood glucose, and compared with teens who were not given a fish to care for, the fish-skipping teens were more disciplined about checking their own blood glucose levels, which is obviously critical for maintaining their health. All right, we are now going to touch upon food security and climate change associated disease. I think everyone here is probably very familiar of the impact of climate change on human health. And we know that things like drought conditions affect communities. It can even create minor disturbances in livestock, which then strongly affect the accessibility and availability of animal-sourced foods, which decrease opportunities for diversified and nutritious diet and increase the risk of malnutrition in certain societies. Observing cereal and livestock prices are crucial for the early detection of hunger crises coming. But what's the role of the veterinarian in helping actually prevent climate change and not just dealing with the sequelae? Because they really seem like unrelated fields. But let me introduce you virtually to Dr. Dipti Pita, who is associate professor of ruminant nutrition at Penn Vet's New Bolton Center. Her research is world-renowned, and Dipti received a $500,000 grant in 2018 and then a $995,000 grant in 2022, which are huge numbers for a veterinary researcher. And one of her primary areas of research is nutrition and methane production. We know what methane production does for climate change. And Dipti found that differences in methane emissions were accompanied by differences in microbial populations as well as their fermentation pathways. And although she focused on dairy cows, the outcome of her projects can easily be applied to other ruminant livestock like beef, cattle, and sheep. Livestock, particularly cattle, produce 25% of the United States methane. A quarter of the percent of the United States methane comes from cows. And methane is a greenhouse gas 28 times more potent than carbon dioxide. Reducing these emissions is critical. And on top of the environmental benefit, reducing methane emissions could increase milk production and animal growth. Dr. Pita has found that low methane emitters are more efficient cows because methane formation is an energy inefficient process. So reducing methane production gives that energy back to the cow, who can then use it for metabolic activities, including improved growth rate and improved milk production. Currently, the most commonly used method for limiting methane emissions is to feed cattle methane inhibitors that prevent rumen microbes from producing methane. But little's known about how these inhibitors affect the microbes. An alternative approach would be to breed animals that naturally emit less methane. Cows naturally vary in how much methane they emit, and prior studies have suggested that this variation is partially heritable, which was a very novel idea. Dr. Pita wanted to investigate whether any host genetic components or other host parameters like the microbiome are associated with cows that produce less methane, because knowing these characteristics could enable us to selectively breed low methane emitting cattle. To do this, her team first identified 5 low methane emitting cows and 5 high methane emitting cows from a herd of 130 lactating Holstein cows. Then she set out to characterize the differences between these low and high emitters in terms of their genetics, milk production, rumen fermentation, and rumen microbiomes. On average, the low emitters produced approximately 22% less methane than the high emitters. Overall, there was no difference between the low and high emitters in terms of food intake, amount of milk produced, or milk composition, although low methane emitters did digest less of the food they consumed. They did find that there were major differences in the rumen microbiome, microbes, and fermentation patterns of the low and high methane emitting cows because methane is produced via microbial fermentation. Low methane emitting cows housed fewer types of microbes in their rumen, and their microbes were less likely to be methane producers. When the researchers compared the gene expression of ruminal microbes in low versus high emitters, basically a measure of how often the microbes are using these genes, they found that the low emitters had lower levels of methyl-CoM reductase, an enzyme that is involved in methane formation. Interestingly, these low methane emitting cows also tended to have smaller statures than high emitters. While the connection between body size and methane emissions is not immediately obvious, Dr. Pita believes it basically comes down to stomach size and the turnover of food within the rumen. There's a bit of crosstalk between the host and the microbiome in terms of whether the host is driving the microbiome or the microbiome is driving the host. Smaller cows have smaller rumens, which means that they can eat less food at a given time. This means there's faster passage rate of food through the gut, and the faster pace is less hospitable to the kinds of microbes that make methane. Now, Dr. Pita is investigating whether it's possible to selectively breed dairy cows to have efficient microbiomes, and these results could be combined with other management strategies, such as the feeding of the methane inhibitors to low methane emitting cows. This is hugely critical work. to reducing some of these gases. So at Penn Vet, hopefully I've kind of shown you how deeply entrenched this research and the One Health movement is. And we actually offer several dual degree programs for students coming through in order to hopefully encourage students to go into these fields. We actually offer a dual veterinary and nursing program that will hopefully be able to send people out into areas that need help, and they can provide both animal support and human support. We have a combined veterinary and masters of social work, a combined veterinary and masters in environmental studies, masters in business association, master of law, masters of education, combined degree with the masters in public health, which is considered our One Health track. And then, of course, we have a combined VMD PhD program. Here at the end, I just wanted to introduce you to one of our former students. In 2016, this is Dr. Brianna Parsons. When she was a student, she was a second-year veterinary student, and she decided she was going to start a goat dairy in the Gambia. The Gambia Goat Dairy, which is now called Fair Farms Gambia, was the first commercial goat milking facility in the Gambia. They aim to achieve food security through environmentally sustainable interventions. Dr. Parsons saw, as a veterinary student, that the Gambia experiences a 10.5 acute malnutrition rate prevalence, and over 25% of its children under the age of 5 are severely or moderately stunted. Livestock are 30 times less productive than those in more developed nations, and the country relies heavily on dairy imports. Fair Farms Gambia is built on the principles of social entrepreneurship, a business model oriented to positively address environmental and societal issues. Fair Farms provides the local community access to affordable, nutritious animal protein foods while sharing best practices for creating agricultural enterprises of their own. That is it. Thank you all for listening. I have to give a huge thanks to all of these people, especially Dr. Jenny Punt, who is our Associate Dean of One Health. And these people all contributed to this presentation today. This is my email, if anybody would like to contact me via email. And otherwise, I am happy to take questions. And you can post questions in either the Q&A or the chat.
Great. Thank you, Dr. Arbittier, for that wonderful presentation. I think it was very, very informative, a lot of information that you shared. And I think it's really a topic which all of us need to be, more aware of, and all healthcare professionals. So I think it's a very timely topic. So we are getting some questions in, and we'll just start with the first one. What are your thoughts on disease surveillance in wildlife to better identify and predict potential human pathogens circulating in the wild?
This is critical. And this is really the goal of One Health, right? Like, we want to prevent these infections before they spill into the human population. And so surveying them in wildlife, we know that wildlife can then either be transmitted directly to humans or then transmit diseases to production animals that then transmit diseases to humans. And so this surveillance is critical, and it's hard to find the funding for that. But now, in response to the COVID pandemic, one of the things that everybody recognizes is that this could have been caught much earlier, and we could have seen that there was this virus that was circulating in the wildlife that was very similar to other viruses affecting humans. And so the wildlife surveillance is huge. And there are many global movements that are working on this. But of course, funding is always a really big problem. But that is the key to identifying emerging threats and then being able to stop them once they begin to spill over.
Great. Thank you for that answer. So just another question about, how maybe students and early career professionals can get involved in the One Health movement and contribute to the field of veterinary health and its broader implications.
It's a great question. Our students at Penn Vet actually can get involved their first year.
Mhm.
So we actually have now created a One Health major for the students. So even students who are not doing a dual degree program can be a One Health major. So we have our small animal majors, our equine majors, our food animal majors, and now we actually have a One Health major. So these students can actually track their veterinary career aimed at service in a One Health profession. And the amazing thing about a veterinary degree that we talk about so much is it is about so much more than treating animals, right? We have people that come to our school who are never going to clinically treat a companion animal. Their entire goal is public health. And so students can get involved in research projects. Our students have the first 2 summers off after their first and second year. They frequently can travel. We have programs all over the world where they can travel. They can work in labs. So our students can get involved very, very early when they are interested. And I think that our school does select for students who, in terms of students applying, for students who have interest in this realm, because it is such a big focus at Penn Vet. We are a very heavily research-based school.
So, Dr. Arbittier, would there be opportunity? I mean, of course, there is opportunity there for students who are joining, PenWet or who would like to, but would there be opportunity for students who are just interested in the topic, maybe to do electives or do an internship or maybe even international students? Do you know if that is a possibility there?
Yes, absolutely. So our students have quite a bit of time to do externships, which is how they do a lot of travel around the world. We have programs in Africa, China, Thailand, just to name a few. And so all of those things, international students can also come and do externships with us. They can come work in research labs, sometimes for credit, sometimes not for credit. So there is unlimited opportunity. And so many of our things, the reason I highlighted Dr. Brianna Parsons is this was a student who just had an idea and she was aware of the nutritional problems and the malnutrition in the Gambia. And she created this thing. There was no program in the Gambia. She found a faculty member, our faculty are very excited to get involved. And she found a faculty member to help her. And she created this program. And so the students can absolutely get involved with whatever they can get involved in. And if Penn Vet doesn't have a program, certainly we talk to different programs around the world and can always figure out ways to get our students there.
Mhm. Okay, so another question, coming out is, can you comment on the economic aspects of One Health, which would be quite huge, I would think, but maybe you can comment a little bit more on that.
Yeah. So in each, that slide that I kept showing with all of the challenges that can benefit from a One Health perspective, all of those things have massive economic implications when it comes to disease. So antimicrobial resistance, not only do you have all of the cost on the human healthcare, you have the human lives, right? 5 million people in the United States died from antimicrobial resistant diseases. And so you have the economic impact of treating these patients, of the studies with climate change and the crops being devastated with either too much drought, too much rain. The economic impacts are everywhere you look. There's not a single thing we look at with zoonoses. All of these governmental agencies cost money to fund. All of the COVID things, they cost money to fund. So in every single aspect of our challenges that we discussed, you could spend hours talking about the economic impact. There is nowhere that there is not a huge economic impact. You know, Look at I spent quite a bit of time talking about the methane production, right? Because methane production is one of the leading causes of climate change, and climate change has a huge impact on everything. So, yeah, the economic implications are massive.
So, Dr. Arbittier, let me ask you this question. Like, of course, you spoke about outbreaks and how things happen. Because we are living now in close contact. Of course, the U.S. is very advanced. There's a lot happening. In the One Health and also in veterinary medicine. But what about like low and middle income countries? A lot of our population obviously is living in those countries, and a lot probably is happening there, but we are not aware or it's not being caught. in time. So what are your thoughts about that? How can that be improved? Or, and I would also be interested in knowing how, like whatever is happening in different parts of the world, how is that fed, or how do you get that information? Because again, it might reach you in the United States also now, since it's really a matter of a few hours that a flight can bring either an animal or a person or a disease, from different parts of the world. So if you could, maybe address those two questions, what are your
Yeah,
thoughts on that?
absolutely. And in all aspects of One Health, um, one of like if you start combing through the research, everybody recognizes that some of these lower income areas are hit the hardest by these things, and the fatalities and the economic impact are often not reported. And they
Yeah,
actually have statistical ways to deal with that because, when you're looking at the CDC coming up with its COVID fatalities, they know that things are underreported. And
right.
these are the areas that are absolutely devastated. And so I think it is very well recognized in every paper that you pull. They talk about how those areas are the ones that are the hardest hit.
Mhm.
And all we can do is keep sending people and not ignoring the data that comes out, right? So we know that when these diseases spread, they are going to spread like wildfire through those communities faster than they're going to spread through, the United States for sure, because they just
Right,
don't have the resources.
right.
And so a huge amount of the research is looking at those populations of people, and it is well recognized. And I think there are some great websites that an easy Google search will show you, to show you some of the global services and groups that are out there. This talk was so hard to get down to 45 or 50 minutes, but I have I have like 50 hidden slides, right,
Right.
of groups that are out there doing the global work. And so the groups are there. Everybody recognizes that those marginalized areas are the hardest hit and the most devastated by these impacts.
Sure, sure. And how does, again, just, maybe the surveillance, or how does these outbreaks happening in other countries, do they reach you at all, or how does that work in the animal world?
So they're reported, and once they get reported through like the World Health Organization or one of the biggies, right? The
Mhm.
UN, the World Health Organization, the CDC, once they start reporting it, then very frequently researchers from those countries or adjacent countries who are intimately involved in those outbreaks then start
Right.
reaching out to the other universities. And so they'll start reaching out to figure out tests, serological tests, work with the different veterinary schools. We have this colony of bats, we want to test them frequently. Body like their carcasses will be sent for pathologists in the United States to work on. And so there is a very tight community in terms of that.
Mhm. So, Dr. Arbittier, any thoughts on how, we can bring this concept of One Health more to the population, to the communities? I mean, of course, even to our healthcare professionals, but it's so important, to bring it to the community. Any thoughts on that? What is, a way that we can bring, material which is digestible, where they can understand these concepts and for them to really understand how it can actually be impacting their health?
A lot of these global groups are the ones that are doing that. They are in these communities.
Right.
They understand the needs of that particular community. Those communities are not interested in all of the bench lab work that's happening at UPenn, right? Like they're not interested in that. They need to know how to keep their cows alive and their goats alive so that they can have milk and they can have animal protein. And so a lot of these global groups are the ones with boots on the ground in these communities disseminating the information, and they need volunteers. Anybody who wants to get involved, they need volunteers to go on these trips and help disseminate these information. And actually, with our dual degrees, like particularly our veterinary nursing dual degree program, these are people that are going to be able to go into these communities, help vaccinate humans as well as animals, work on animal productivity, and also deal with some of the malnutrition and things like that. So these global groups are out there. They are just like woefully understaffed. And so anybody who has an interest, you can spend a little bit of time volunteering with these groups pretty much in any continent and have a big impact. They, you know,
Right.
they need the help, and there are a lot of groups out there doing really good work.
Great. Dr. Arbittier, maybe just the last couple of questions. So what are your thoughts on, there's a lot of talk, of course, about GMOs, right? Genetically modified food for human consumption. In a similar manner, is there genetically modified feed available for animals? What are your thoughts about that? Is it affecting their health? And then sort of, maybe even causing transmission of disease, or what are your thoughts on that?
That's a really good question. And I'm trying to think, I'm not sure about genetically modified in terms of food, in terms of like grains and things that are being fed to cattle. You know, that's kind of what I think about in chickens and pigs.
Right.
I don't know the answer to that. I do know that a lot of research is going on to look at using waste, like food waste products to feed to these animals in order to create like a more sustainable food chain. But in terms of genetically modified feed, I'm not up to date on that.
Right.
Um, there is a lot of research going on, and actually, Dr. Pita, who I mentioned, has a study right now looking at all farmers who have produce that they can't sell because it's damaged or it's too old or whatever. These are tractor trailer tons of fruits and vegetables that they can't sell. And so she's looking at repurposing them into production animal feed and looking at the digestibility and what it does to the microbiome of the cows and things like that. So that kind of cycle to keep things more sustainable is out there.
Okay, so just maybe the last question. I know we are beyond the hour for you. So I was just curious about, of course, we hear a lot about, disease being transmitted from animals or birds into the humans, right? And of course, in certain cases, it just stops, the transmission doesn't go on. And do you see a lot of it happening the other way, meaning, disease transmission from humans to animals? Do you see that in practice, or is that not as much of a concern?
That is an interesting question. It's not as much of a concern, but I will tell you that during COVID, humans were getting COVID, and cats, it turns out, are particularly susceptible to COVID more so than dogs. And so people who had elderly cats at home, like their beloved elderly pets, the cats were
Right.
getting COVID and dying from the
Oh,
humans. And
oh,
so that is actually really interesting. Additionally, it was seen quite a few zoos identified it in their large cats, like their tigers and their lions.
okay.
Um, those animals showed symptoms of disease, in general, didn't die. So,
Mhm.
what we found during COVID-19 is that if you have an immune compromised pet, I'm definitely aware of a couple of dogs that died as well, but particularly cats and hamsters. Hamsters are very susceptible to COVID-19. You have to be really careful handling your animal because if they are immune suppressed for any reason, then they are much more likely to succumb.
Great. So I think that's it from here. But I think it's been a fantastic session. Of course, I think there's a lot to learn from everyone. And again, for all of us in different healthcare professions to really be learning from each other and then taking it back to the communities. So really, really grateful to you. And thank you for taking time out from your busy schedule for doing this for us, for our healthcare professionals. So really grateful. Thank you so much, Les.
Oh, thrilled to be here. Thank you so much for having me. It's really been an honor.
So thank you, everyone, and assalamu alaikum, and good evening, good night, wherever you are. Thank you very much, Dr. Arbittier, again.
My
Bye
pleasure.
bye
Thank
for
you.
now.